The following content is provided by MIT OpenCourseware under a Creative Commons license. Additional information about our license and MIT OpenCourseware in general is available at ocw.mit.edu.
Okay. Welcome to the second discussion of networks and the second to last talk of lecture of this course. There's a very important set of lectures after that from the students I'm looking forward to. So last time we talked about the use of computers to model various complicated systems in mainly cellular systems, ranging from highly cooperative, even bistable bifurcations, that sort of where a cell makes a decision, a discussion of chromosome copy number and its implications, and a large chunk of time on flux balance optimization, where we can look at many of these things from the standpoint of optimization. Now, today, to paraphrase a famous local politician, we will ask not what computers can do for biology, but for what biology can do for computers. And of course, we'll go back and forth and try to look at what kind of this interesting dynamic between the experimental and the computational side. And so how can biology aid algorithm development to return the favor and aid biology? But not only algorithm development, not only inspiring algorithm development, but actually implementing hardware and software in biological systems. We'll talk about that. And ranging from molecular computing to cellular computing, and then back to inspiring algorithms again. So, slide 3, what is it that we're really talking about that computation needs in terms of aid? How do we get, what are the real issues here? And we've mentioned a couple of times in the course, probably initially when we were talking about dynamic programming and related topics, of how a problem in computer science scales. Typically you'll have one key input size, n, and then the running time, or sometimes the memory, has an upper bound, which we've been referring to as the order of some function of that n, where n is the length of a string or the size of the problem. There are other symbols that are slightly more rarely used. There's, in addition to the upper bound, there's a lower bound. Sometimes you can get an exact bound or equal bound. And how does this play out when you have specific instances of n? Now, of course, when you say that something's on the order of or upper bound of a function of n, you don't always necessarily describe it with constants. But to give you a ballpark, when you have n ranging from 1 to 1,000, and the polynomial ranging from linear to quadratic to 10th power, and then the exponential or factorial. And you can see that for very small n, you can get cases where a polynomial performance, computation time, can be longer than an exponential computation time. So exponential isn't necessarily bad news depending on the size of your problem. But it gets to be bad news very quickly. As n increases, you quickly get over large numbers on the far right-hand side that are computationally intractable with any known computer. So we mentioned computational complexity as one of the various definitions of complexity at the beginning of the course. This is not the one we chose to be closest to the definition of living complexity, but it's one that's very frequently used in the computer science field. And I just want to introduce a few of the terms here, just so you've kind of heard them in this context. We have, and it basically refers to the issues brought up in the previous slide of whether it scales by a polynomial, which is generally desirable, or whether it scales exponentially or worse. And so we have P are problems that we can solve in polynomial deterministic time. Deterministic just describes the fact that the algorithms will do the same thing time after time, which is typical, the sort of typical computer that we feel comfortable using. And an example of this, of problems in the P class of dynamic programming, which we've used numerous times. It scales polynomial, and the polynomial just depends on the problem slightly, from squared to the sixth power, we've seen. NP has a number of subsets, but overall it means it's non-deterministic polynomial time to get the solutions. The solutions are checkable in polynomial time, but are generally felt to not be currently feasible in polynomial time for the determination as opposed to checking. An example of this where actually the inventors of various encryption schemes, such as RSA, the R and the S and the A referring to the last names of the authors, in a certain sense they're banking on the difficulty of cracking these codes in polynomial time. You can use the codes if you know it. You can check it, that is to say, but you can't crack it unless someone comes through with a breakthrough on the NP problems. Because if you solve one of them, you can solve all of them. And then there's subsets of this. This is a little less critical for today's discussion, but there's NP complete, which an example of which is sort of the traveling salesman. Can you get through all the vertices on the trip with a mileage below sort of some threshold? And an NP hard version of that same thing is what's the minimum mileage that you can get, not just less than X, but how much less than X. And the worst case scenario is undecidable, where you really, even given an unlimited amount of time and space, you can't tell whether it is. And the classic one is the program halting problem, where you don't know whether your program is going to halt. And probably all of you have run into that problem. I'm just being funny. But I mean, it is the program halting problem is a serious mathematical construct. Okay, how do we deal with this? How do we start thinking about ways that we've dealt with it before and ways that biology could change the landscape a little bit? Usually, what we do when we're faced with an NP hard problem is cheat in some way or another. You redefine the problem so it's in class P, sometimes sacrificing something. So you might have, if you're interested in tertiary structure, you may redefine it as secondary. And we showed that secondary structure for RNAs, it can be solved with a dynamic programming algorithm with an n squared or at worst n to the sixth algorithm. Whether that is as precise as the most precise tertiary structure that one could get given sort of infinite exponential time is an open question. Probably not. If n is small enough, we showed that exponential times can be reasonable, and so you just do an exhaustive search. Or if you can't do that, then you use some clever heuristic way of pruning things. And that's, in a certain sense, that's what most of the approximations are. So what can biology do? We'll talk about three examples today. One is DNA computing, one is genetic algorithms, and neural networks. The first one, none of these really actually solve the problem. The first one is a way of just obtaining a lot more raw computing power. I'll show you a quote where they say they have solved an NP-complete problem, but it's in the same sense that you can solve any exponential problem by brute force. That's not really finessing it out of NP into P. Genetic algorithms and neural networks are definitely heuristics. They're beautifully inspired by two of the greatest algorithms in the history of life on Earth, and that is evolution and complex brain networks. And so genetic algorithms is based on the adaptation that occurs during evolution and recombination and mutagenesis, and neural network is also about adaptation, but this is on sort of the time scale of learning. So we'll first dedicate ourselves to molecular computing and associated, just kind of put it in the context of nanocomputing in general. And you can see all the issues in computing, not just the math module. So all the steps are assembly of the requisite hardware. This is some kind of factory operation, typically. Then there's some sort of input module, some hardware and software that's required for getting the data in. Then there's some sort of memory. And then there's a central processor, might have math components and output. This is sort of like from our first lecture. Assembly, input, memory, process, and output. And what we want to do with biology is we want to harvest things from genomics and from just biological research in general to use to design better computers either in silico or in a biochemical sense. And then harness evolution either to make devices or as part of algorithm development. We have different people have different opinions about how much longer Moore's law, the scaling for large-scale integrated circuits, or Kurzweil's version that goes back to 1900, about doubling every 2 years of the ability to calculations per second per $1,000. There might be another decade left in the silicon large-scale integrated circuit. That's what some people say, or maybe more, maybe less. So there are three real options here for that next step: electronic nanocomputing, optical nanocomputing, molecular nanocomputing, and you could add to this quantum computing for maybe 4 different options for beating Moore's law or extending Moore's law, depending how you look at it. So let's just walk through them quickly, one at a time. Optical computing, you can think of as already here in a sense, in that our optical fiber networks have very fast switches that are required for a good deal of our fastest internet. And there are various demonstrations of where you can do optical computing for many of the tasks, not just the data transfer. And just like many other things, there's a desire to shrink this down for cost of manufacturing and quality and so forth. The advantages of the optical over electronic, typical computers, is that for a given set of operations, there's the general sentiment that it might be lower heat generation. It goes at the speed of light rather than the slightly lower than speed of light that typically comes into actual implementations in electronic circuits. And here's 2 examples taken from the literature of getting natural sort of self-assembly, just as we've seen in many biological systems where you get self-assembly of membranes, self-assembly of multi-protein complexes. Here you want to make particular kind of size of optical particles that have the right refractive index and spacing and shape and so forth. And you can use self-assembly here for that sort of thing. These are examples of where we're getting sort of in the nanometer range here. This is a 5-micron scale object here. I've chosen this particular example. There are a number of examples of electronic nanocomputing where the electronics is getting down to the sort of size of molecules. Here the molecule chosen is a polymer of carbon, not a hydrocarbon, but carbon. It's like the buckyballs, which is carbon-60, or graphite rolled up into tubes. These nanotubes can be used as transistor-like elements in very tiny circuits. This is just a schematic. This isn't actually a micrograph, right? And here are 4 circuits that kind of reflect what that might be your first 4 projects in an introductory electronics course, ignoring the fact that you might not use nanotubes. But you would have voltage in and voltage out. In other words, this is a transistor-like circuit or an inverter-like circuit in the upper left-hand corner of slide 10. And the nanotube here is in the middle in series with a resistor going from high voltage to the ground at the lower part, and the voltage in essentially modulates the voltage out in a nonlinear curve, and you can see this highly cooperative curve, just like the ones that we've been talking about in a number of other biological and physical systems. The upper right, you have a NOR gate. Almost every circuit can be made by combination, simply by not ORs. This is an inverter plus an OR. And so you can see that they now have two inputs on the left and right in 1 and in 2, and they can have states 1, 1, 1, 0, 0, 1, 0, 0 going from left to right. And you can see only when they're both 0 does the whole circuit output go down to 0 or low voltage for output. So input, there are 3 possible input combinations, and the last one gives the low. And this is all done with these kind of molecular scale nano tubes. Here's a RAM. It fires 2 nanotubes in order to store, where you flip it from open to flip it from low to high. In the last example in the lower right, you now need 3 nanotubes. You see we worked our way up in complexity from 1 to 2 to 3. And you need 3 in order to get a ring oscillator. That's the way you would think about, most easily think about it, where you have the first one's output affecting the second one affecting the third, and the third one looping back to the first. And the result of this is you get a series of peaks and troughs here, which you use for synchronizing circuits or generating other useful sinusoidal processes. Now that's one example of sort of optical electronic. Molecular includes a number of different possibilities, including DNA, which is what we'll focus on, DNA computing. And this was started by a physicist famous for thinking out of the box quite a bit. Feynman, in 1959, when he was still fairly young, gave a talk entitled "There's Plenty of Room at the Bottom," and by that he meant that we can, just as we can machine and manufacture objects under fairly automated fashion, we should be able to scale that down to the point where we're manipulating individual atoms. And he couldn't think of any physical reason, such as the uncertainty principle or anything like that would prevent one from doing that, manipulating individual atoms. And many years later, now he's been proven right in that we are doing that, albeit not in any really high production method. Drexler, in his thesis, and subsequently has championed, is probably the right word, this sort of notion in given it the name nanotechnology and nanosystems, and really fleshed out some of the things that one might be able to do if you had a higher throughput way of dealing with manufacturing at atomic scale. However, even he did not really connect all the dots between where we are now and how we get to the very first nanoassemblers and nanotechnology. Since then, there's been kind of a renaissance of interest in this, with the recognition that biological systems actually are sort of naturally doing nanotech scale atomic manipulations, and you've seen a few examples of that in this course. But the particular instance that we'll use as a jumping off point for discussion of all the steps that could be where biology and molecular mining could give us new tools, whether it's from assembly, input, memory, computation, output. We'll start with Lynn Adelman's pace-changing paper in 1994. This is the A of the RSA that we talked about just a few minutes ago. He was obviously a hardcore, and is, algorithmics expert, and decided in 1994 to actually do a paper that required not only algorithmics, but a huge change in the way you implemented the algorithms, and then actually to go into a biochemical laboratory, for which he was not previously trained, and to author a single author paper that included such things as PCR. That was in 1994. There was no literature on the subject of DNA computers before that. A mere 6 years later, there were 520 references on the subject, so he obviously hit some kind of nerve. First few years of that, after that, were mainly theoretical, but I'll show you some examples. But his paper had an experimental component, and some others that I'll show you. Since this course is really about that interface, constantly checking the theory with reality, those are the ones that I'll emphasize here. So the question that he asked in 1994, and is still fresh today, is there a Hamiltonian path through all the nodes in a network? So we've been talking about interesting biological networks, but here, just in any network where the black nodes are connected by directed edges in this directed graph, you want to go from the start S to the terminus T, from 1 to 6, obeying the arrows and going through every point once. How do you do this? And how do you do it in DNA? So an example of one here is going from S to 3 to 5 to 2 to 4 to T. Okay, so the way you do it, first in broad strokes, you encode the graph, both the nodes, black spots, and the edges into single-stranded DNA sequences. Then you create all possible paths by using overlapping sequences to indicate which node is connected to which other node by an edge in which direction. So you can actually have directionality just because DNA has directionality. And you use DNA hybridization now to do that step. Now the first step is linear. Encoding the graph is linear with the number of points in the graph, the number of places in the Hamiltonian path. The second step is not something, and that you would do by having your computer program a DNA synthesizer, which is an automated machine that we described a couple times. But the second step is out of your hands. This is something that happens automatically when you put DNA in the solution. And if you design these sequences carefully so they don't cross-hybridize very much, then the only way they can assemble is the way you want it. And then finally you determine whether a solution exists, and this is something which is almost constant in complexity. So the entire thing scales very gracefully instead of scaling exponentially as the Hamiltonian path problem normally would. This gives the appearance of scaling linearly in time, which is really one of the best case scenarios for polynomial time, and certainly better than exponential. So how do we actually do that? That was broad strokes. This is a more detailed view, and you can see how this really seems like it's going to work. You have each of the nodes encoded by sequences, let's say red and tan here on the top left of slide 14. And you have, if you want to connect node 3 with node 4, so that you take the right-hand end, the 3' end, which is sort of greenish tan, and you connect it now to the other end of, that is to say, the 5' end of node 4, just below it, which is blue. So now you have this hybrid, which is ordered, so it's an arrow going from 3 to 4, and that edge has this particular sequence going from 5' to 3'. Now in practice, you want the edges to be complementary, not identical to the nodes, and so all the nodes are actually represented as reverse sequences in the lower left-hand part of slide 14. And so represent all the edges that connect nodes in this directional manner, and then an example of how you would connect 3 nodes, 3 to 4 to 5, by 2 connecting directional edges, 3, 4, 4, 5, is shown here. All the nodes are in reverse complements, and all the edges are in the forward direction, as arbitrarily defined here. And you can see how they stitch these together, and you make firm connections that are unambiguous, non-cross-reacting, and have a directionality. Now you're starting to get the idea that now we can encode this in DNA, but how are we going to actually do the computation, and how are we going to find out who the winner is? So what is done, remember we want to create all the paths, and then ask whether any of them go through all the points, and then do any of them go through all the points exactly once? So the first thing is to create all the paths from start to terminus. And by just throwing in this mixture of all the edges and all the nodes, you'll create, in principle, all the paths. Now you want to, right, you'll go from the prefix of one into the suffix of the other in the same way that illustrated on the previous slide. And here are some examples of some of the paths. Some are very short. This only goes through 1, 2, 4, 6. Only goes through 4 of the nodes. That's not all 6 of them. The bottom one goes through too many nodes, and some of them it's going through repetitively. But you get the idea, is that you can define the path in terms of all these edges and the reverse complements, which represent the nodes. But here's the actual algorithm as encoded in DNA and implemented by practical methods that a computer scientist can do without too much help, at least not enough help that required co-authorship. So we've already encoded the graph in the DNA sequences, and this is done by automated oligonucleotide synthesis. You create all the paths from S to T by PCR amplifying from the S end of the oligo to the T end. So that means the mere fact that they PCR amplify means they must contain node 1 and 6, the start and terminus. So that's good. Now you want to get the ones that visit every node. So by serial hybridization, you can have nodes 2, 3, 4, and 5 immobilized, and you bind the PCR products to it, and then you elute from 2, and then in series you bind it to 3, elute from 3, bind it to 4, elute to 4 or 5. So now you know it has 1 and 6 because that's the PCR primers. It has 2, 3, 4, and 5 because it bound to them in series by hybridization. But that you could get some of those long paths that went through multiple nodes multiple times. If you wanted to have exactly n nodes, then what you do is some sort of electrophoretic sizing, and here, if you have a calibration curve as shown at the bottom of slide 16, we have known DNA size markers and known PCR products going, showing that you have one of these DNA nodes, 2, 3, all the way up to 6. And if you find a solution that has all these properties, PCRs, does serial hybridization to all the nodes, and is the right length, such as the one in column 6, then you know you've got a solution. And that was Lynn Edelman's argument that he had DNA computing working. 6 years later, we now have, or as of 6 years later, there are now over 500 examples of this. I'll show you this example as both as an introduction to the satisfiability problem and also as showing that you can do RNA computing and that you can encode two-dimensional objects, and it illustrates a number of things. The problem here is a chess problem. Very simple chessboard. It's not 8 by 8, but 3 by 3, and you've got an artificial number of knights here. And those of you who know chess know that these knights can attack in a curious combination of straight and diagonal. And it doesn't really matter. The point is that there are a variety of arrangements of any number of knights such that none of them can attack each other, and they're all kind of at peace here. And the object of this algorithm is to find those combinations. And you do it by cloning. So that was something that was not in the previous example. By cloning, you could find each of the solutions, and you then determine what is present in cis along that clone. It's kind of like haplotyping or splice form analysis. You can really only analyze these things by looking at the product of a single molecule, and that's what cloning is about. It's looking at amplifying that single molecule up to the point where you can analyze it. So that's one thing that's new. The other thing that's new is you start with an RNA in order that you can use this powerful method, this enzyme called RNase H, which has the property that when you bind a DNA oligonucleotide to an RNA and they're complementary, RNase H will destroy the RNA at that point, at the point of hybridization. So it's a way of eliminating an entire molecule if it happens to have a particular sequence in it. And so one of the ways that you can ask logical questions about each molecule in a large, complex mixture of molecules is using this RNase H elimination. And in a way, it's a way of designing an infinite number of restriction enzymes. The RNase plus DNA oligonucleotide provides, in a certain sense, a custom restriction enzyme. In any case, the other thing that's unusual here is the idea of using split and pool oligonucleotide synthesis. We introduced this in the lecture where we were introducing drug protein interaction and ways of synthesizing drugs and other molecules by pool synthesis. And the idea behind split pool synthesis in this case is that for each of the squares in this 3x3 matrix can have two states, either has a night or it doesn't. Each of those two states you can consider a 0 or 1. You can represent them as two different sequences, sequence A or sequence A prime, representing presence or absence of something. And so you have you have 9 squares, and so you have 2 to the 9th different possibilities. And so down below, they synthesized a set of polymers where you have every possible binary state for this 3x3 grid. And that's done by you're synthesizing along, and you come to where you're going to synthesize either A or A prime. You split it, half of this pool gets A, half of it gets sequence A prime. Pool them back together, half gets B, half gets B prime. Pool them and split them and C prime, so forth, all the way out. You really only need 9 of these. They did 10 for some reason or other. But the point is, to get 2 to the 9th power, you need 9 of these. And then, you can read them out electrophoretically, just as the electrophoretic readout gave a sizing in the first DNA computing. You can use it to get the sizes of these. And you can see, you can read out this here's 2 solutions, B, E, F, H, referring to the squares on the 3x3 grid, and E, F, C, these are both solutions. And B, E, F, H, the way of reading this off this combinatorial synthesis from the bottom is you've got A through I as the possible bit binary signatures, and then you have 2 columns, either the 0 column or the 1, the 2 states, the 2 sequences. And as you PCR from the N tag out to each of these tags, A or A prime, B or B prime, and so forth, then you get, these are all, you'll get this graduated series. It'll tell you you've got A is in the 0 state, B is in the 1 state, C and D are in the 0 state, E is in the 1 state, so forth. And so I've circled B being in the 1 state, showing that there's a night in that position, in position B. And you can go through the same thing for the other solutions. Each of these just developed as a clone. And the neat thing about all these problems is they have multiple representations. You can represent them in DNA, you can represent them in data, you can represent them as a Boolean logical set of operations where these things represent ands and ors at the bottom of the slide, and so on. And you will see that in the last examples, where I'll just kind of breeze through, where that set of logical operations is probably one of the favorite points of attack for DNA computing still today. So what are the problems and the advantages? The problems are that yes, it is polynomial time. In fact, it's close to linear time with a number of inputs in terms of synthesis needs. Your computer tells the synthesizer what to make, and then enzymes or hybridization will do this highly parallel reaction independent of n. So basically constant time. So it's linear time for synthesis, constant time for the computing, and constant time for the getting the answer read out. But you have exponential volumes. For example, a 100-node graph, we've been talking about the various graph solutions, might be 10 to the 30th molecules. And if any of you have ever tried to synthesize a mole or 10 to the 24th molecules, you realize it would bankrupt our planet to make 10 to the 30th molecules. So, in addition, the elementary steps are slow. Okay, it's highly parallel. You can imagine having trillions of molecules computing in parallel, but the elementary steps of hybridization and DNA polymerase or RNase H and so forth typically are in the millihertz range. That is to say, it might take 1,000 seconds rather than gigahertz, which would be a billionth of a second. So there might be a 10 to the 12th gap in rate of executing the commands, but there's more than, the hope is that there's more than a 10 to 12th advantage in parallelism. In addition, experimental errors mustn't be swept under the rug. You've got issues with mismatch. There's a limit to just how cleverly you can design all these sequences. As the graph gets bigger, you need to have more and more sequences involved, and that it means that you're going to get more and more cross-hybridization and complete cleavage and so forth. There are, when this slide 18 says non-reusable, there are reusable forms, and we'll get to those in just a minute. So those are the disadvantages. What are the promises or the possible advantages? High parallelism could be much more than the 10 to the 12th fold loss in speed. When computer hardware and people dream of the next generation of computers, they hope to get away from the current record, which is around 10 to the 9th operations per joule for conventional computers. Maybe that's not a record, but it's conventional computers. Closer to the 34 times 10 to the 19th operations per joule, you should be able to squeak out near the thermodynamic limit. And as it turns out, many DNA enzymes, such as DNA polymerase, are already within a factor of 10 of that goal, while conventional computers are off by 10 factors of 10 or more. If one can, quote, solve, or one NP complete solve problem, you can get many. And the improvements that we'll just kind of briefly talk about that keep people excited about this are that this is a natural way of talking to biological problems. If there are biological problems, you can it may be a smaller step to get to DNA computing, and there are faster readout methods, just as there are faster and faster computational methods. And natural selection, evolution, is something that you can use on DNA computers, which so far has not been extremely powerful in conventional computers, although we will talk about genetic algorithms shortly. So one way of getting reuse is to have a so-called sticker-based model, or something where you're basically just using hybridization properties without being destructive. And here's an example. I'm not going to walk through it too much. I should point out the et al. In here includes Adelman again, and I'll have one more example of his work in just a moment here. But there are examples now of work trying to consider seriously the volumes of DNA that are needed, and ways of dealing with fault tolerance or error reduction algorithms, where you actually go through and consciously say, okay, if we had an error here, how would we compensate for it? How would we dedicate a few more bits to take the next step? So, just like in the Knight chess problem that we had a little bit before, the idea of using ands and ors of Boolean variables, these x's in slide 21, which can have two states, 0 or 1, similar to the Boolean variables that we run across from time to time in genetic models. And you can have clauses, which are basically logical operations on the set of Boolean variables, where you have nots and ors and ands. Okay, and this kind of problem is a very general problem, a very interesting one, and it has been tackled just in very analogous ways to the ways we've been talking about the previous ones, where you encode the graph in DNA sequences, thereby creating all the paths by hybridization or something like that, and then you read it out with PCR and solid phase. And you can say, you see, here's a quote where they say, here we solve an NP complete problem. And they have indeed solved one, but they haven't really turned it into a polynomial time problem. They simply brute forced it out. And then this is the most recent one, just it came out in Science, again, Lynn Adelman on it, and now up to 20 variables in a 3-stat problem. Okay, so that's all about computing. It may not, since DNA is relatively slow at computing per step, highly parallel, it may not be the best of the various steps in computing: assembly, input, memory, computation, and output. So we'll explore some of the other ones, in particular assembly. To a certain extent, assembling computers is slow, and so it's something where molecular assembly might have some advantages. Now, this particular example, I'm going to emphasize the assembly aspect of it, but you can think of this as a way of mapping. These authors have mapped assembly of an actual two-dimensional tiling onto an abstract but important and powerful computer science concept, which is the general Turing machine, a machine, a kind of a tape-like structure that can do any kind of general computing, deterministic computing. And so they've mapped a kind of a physical tiling, as you might have a periodic or aperiodic mosaic of tiles, onto this computing machine and onto this kind of logical operation XOR on a string of binary bits, just as we had in the previous couple of slides on the 3-stat problem. I'm going to emphasize the geometric physical version of it because we're trying to make a transition now in this paper which combines the DNA computing with the DNA assembly. Now, we have 3 more or less equivalent ways of representing the same sort of sequences. Now, these are called triple crossovers because you can see, let's take a look at, say, Y2 here, right in the middle of the slide. You can see that there are these multiple crossovers where you have a kind of a recombination event where you've got 2 double-stranded DNA molecules that are exchanging a strand. And this is not a natural homologous recombination event in the sense they're non-homologous, and this thing is kind of trapped in this crossover. And the crossover, when you have multiple crossovers, you can make a piece of DNA that now has more than just 2 ends. It can have multiple sticky ends. It can have, say, in this case, in the upper part, you can see 4 different ends with 1, 2, 3, 4 crossover strands here. And each of these, you can see on the far right, a 5 base 3' overhang and a 7 base 5' overhang and some flush ends on the far left. And so each of these things has ability to stick to other tiling elements. And you can see that you can put together a 2-dimensional structure which can be as intricate as a mosaic. It does not have to repeat itself, or it can repeat itself if you want to use visual tools such as Fourier transforms to look at a repeating structure. And you can see you can engineer in restriction sites to help you analyze the structure on a gel-based assay on the far left. We've seen enough gel-based assays tonight already. We're not going to walk you through it, but you can see that there's, even though this is not straight DNA as the previous 2 or 3 examples were, this is a much more complicated branch structure. Nevertheless, you can turn it into a linear readout with electrophoretic sizes, but you can also look at it as a truly 2 or 3-dimensional object here. Here we use they use atomic force microscopy, where a probe with a single atom at its tip is responding to the force that allows that as you touch an object, the feedback in the system with a scanning tunneling microscope, usually as a part of the feedback, tells you that you've just touched the surface and you've got to back off a little bit, and then you scan along and just profiling the surface. And so here, with 2 different tiling methods, this is more a repetitive rather than an aperiodic tiling. You can see that these little pink protrusions, you can engineer into the tile not just the 2-dimensional sticky tags, but a 3rd dimension, which is a bump, which might be a stem loop, as we've seen in other secondary structures. is engineered into this DNA. So now that these bumps will stick up and be easy targets for the atomic force microscope. And here you have a bump every second tile, and that's on the left. And on the right of slide 27, you have a more complicated tiling where you have 4 different types of tiles and a bump every 4th one. And so you can calculate from the Watson-Crick model for DNA or more advanced models of DNA, even though this is a lattice of branch structures, you can calculate it should be about 33 nanometers between the bumps, and that's what's observed, and 65 nanometers is calculated and observed. You can see the bigger lattice spacing in these admittedly somewhat fuzzy atomic force micrographs. But you can see you get something, an experimental confirmation of the two-dimensional structures here. Now, this is self-assembly nanofabrication, and to some extent it is inspired by and can be combined with microfabrication. This is something where you basically use optics. You go to the limit of current optical manufacturing as occurs in microfabrication, where it's typically hard to get below, say, 100 nanometers or so in feature size. Here you can see this is and this is the sort of microfabrication used to make your computer chips, but it's in this case it's used to actually make moving parts that move relative to one another, and that motion actually has useful applications. The first such useful application that I'm aware is putting these things into airbag sensors. And the idea is that when you're driving your large automobile and run into a even larger object, you suddenly decelerate either by brakes or some other method. And when you do, this little bitty device, a sensor inside somewhere in your car, will shift one of its parts by at least 0.2 angstroms, meaning 1/10 of an atomic diameter. That doesn't seem like very much, and it isn't. It only causes 100 femtofarads, that means 10 to the minus 13th farad capacitance change. But that's quite enough to signal that a collision has occurred or will occur, and with a very low false positive rate. Those of you who have driven large automobiles know how infrequently the airbag opens up accidentally. But when it does deflect by 0.2 angstroms, then it does open up the airbag. Okay, so this is a payoff of microfabrication. But now we want to combine the sort of nano systems we saw in the DNA computers and in the tiling with microfabrication. And I picked this as just one of the very few examples where microfab meets nanofabrication. And we'll call it a nanoelectromechanical system. And here, so you've got the microfabrication of both the posts upon which these things stand, which is sort of these nickel columns of 80 nanometers, and the little metal bars, which can be a sort of the micron range, and you can see them visualized in the left-hand photograph, where you have these bars at regular spacings, and if you look at this publication or the webs that go along with it, these little bars will spin around. And what they're spinning on is not a microfabricated motor, but it is a nanobiotech motor, and actually it's a protein that most people didn't think of as a motor when it was first discovered. It is the protein present in almost all organisms that is responsible for ATP generation. Usually you think of this as making ATP for motors to use, but this actually, since it is capable of rotary motion, does move this 1 micron bar around at the rate that you would expect for the sort of nano machine to be generating torque. Okay, so that's we've talked about assembly. That's an example of an output device. What sort of input devices we have that will work at the single molecule level. Here is there are many examples. We mentioned a few of them in the sequencing genotyping lecture, where we were talking about single fluorophores. Here you can use another aspect of biology, which is the self-assembly of membranes, to make a very, very tight, low-conductance seal, which is only on the order of 2 nanometers thick, but it's enough to make a gigaohm seal, a multi-gigaohm seal. And then you poke a little hole in it with a single molecule of a protein. And there are growing ways to do this on inorganic substrates as well. So when you have a single protein pore, which itself might be a 1 nanometer opening, it will allow, in the presence of electric field, indicated by these negative and positive charges here, yellow negative and positive ions to go through, like sodium chloride. And they will go through at about up to a million ions per second easily. And when you have a larger molecule that's say a polyanion in the electric field, it will slowly migrate through this pore, and while it does so, it blocks the rapid movement of the smaller ions like sodium. And you can record the it doesn't necessarily completely block the channel. You can record the rate at which sodium will go through and how it's influenced by the composition of the polymer going through. And so here's an example of a bacterial protein. This is Miller et al., reference down below. A bacterial toxin, actually, from Staphylococcus, whose goal in life is to kill other organisms. It's not to provide a handy conduit for RNA to go through a cell. But nevertheless, in this experiment, when a nucleic acid does go through this, it blocks the little red water molecules and similar sized ions are blocked, and they're blocked in a way which is sensitive to not only the molecule but parts of the molecule. So you can see here you've got a molecule with a oligo A part and an oligo C part, and you can actually discriminate between these, both in terms of the rate at which they go through, each of the parts goes through, and the conductance. So if you look down in the lower right-hand part, you'll see a 5 picoampere to the minus 12 ampere, 20 picoampere, and 120 picoampere levels for these individual molecules. And each of these spikes is first the A30 half and then the C70 half going through, and you can see that the two different conductance levels that you get reproducibly going through. It's going through typically in one direction, first the A and then the C, and different conductance levels, different rates, and then the 120 picoamperes is in between molecules where you're getting the full conductance capability, lots of sodiums going through. And just like with other methods that we've seen before, the use of 2 dimensions helps get you better statistical resolution. Here the 2 dimensions are the conductance and the time on the vertical axis and conductance on the horizontal axis, and you can see how you can discriminate different types of polymers by this method. One molecule at a time. Each of these dots represents a single molecular event. And, so we'll take a short break, and then when we come back, we'll talk about not molecular computing, but designing cellular computers, and revisit some of these same themes. Thanks.